in 2 men and 1 in 3 women are at risk of developing cancer. Each year, 12.7 million people are diagnosed with cancer, and 7.6 million people die from the disease. As I am interested in medicine, and specifically in medicinal plants and how nature can be an inspiration for scientific innovation and medical treatment, I found one candidate for further research: Taxol. This is a natural compound found in the needles and bark of the common Pacific yew tree, Taxus baccata, and is used for treating different types of cancer. This tree is most commonly found in Central and Southern Europe, but nowadays we are also able to make this medication synthetically. So, the research question I'm trying to answer is, how is Taxol effective in treating cancer? To determine the power of Taxol, I collected a sample of the Taxus baccata leaves in a park next to my house in Geneva and tested its ability to inhibit the growth of E. coli bacteria. The inhibitory effect was clear, proving to stop the growth of bacteria. A great first step proving its potency, but not enough to answer my research question. I needed to do the experiment with Taxol on actual cancer cells. Therefore, I contacted Jonne Rijmakers, a postdoctoral fellow working at the National Cancer Institute in the Netherlands and doing research on Taxol. She invited me to conduct my experiment on breast cancer cells using synthetic Taxol in their laboratory. My findings will be presented hereafter. First, I will explain what cancer is, then I will set forth the history of Taxol. After this, I will discuss how Taxol kills cancer cells in 3 sections. How do normal cells divide? How do cancer cells divide? And then how does Taxol interfere with cell division? To then determine the limitations of Taxol, I interviewed Jonne Rijmakers. This interview will precede the conclusion, if Taxol is indeed effective in treating cancer cells. The human body. An immensely complicated anatomy made up of trillions of cells, each with a specific purpose, and each with the ability to divide to continue fulfilling its purpose. But when this simple and crucial process of division goes haywire, damaged or old cells which should die survive, and new cells form uncontrollably when is not necessary. These cells differ from normal cells in many ways that allow them to grow out of control and become invasive. For one, their ability to divide for an eternity is the result of their ignorance towards signals that normally tell cells to stop dividing, or to begin a process of apoptosis, programmed cell death, which the body uses to get rid of unneeded cells. These cells are also often able to evade the immune system, a network of organs, tissues, and specialized cells that protect the body from infections and other conditions. Although the immune system normally removes damaged, abnormal, or dangerous cells from the body, the immune system is not always able to recognize these cells as an enemy because they are able of hiding their chemical ID from their surface. These cells form tumors, which spread into or invade surrounding tissue and nearby organs, where they can cause serious and eventually fatal damage. Not only this, as these tumors grow, some of these cells can break off and travel through the blood or lymph system to find a new location far from the original tumor to form new tumors. This occurs in over 100 related diseases, which is given one collective name: cancer. The different types of cancer are usually named after the organ or tissue of where it originated. For example, brain cancer starts in the cells of the brain. Lung cancer starts in the cells of the lung. When lung cancer, for example, spreads to the brain, the tumor in the brain is still built up of lung cancer cells and not brain cancer cells. This globally known genetic disease is caused by changes to the genes that control the way our cells function, especially its ability to grow and divide. These changes, or mutations, can either be inherited, be an error that occurs during cell division, or be the result of the damage done to our DNA caused by certain environmental exposures, such as chemicals in tobacco smoke, and radiation, such as ultraviolet rays from the sun. Each person's cancer has a unique combination of genetic changes, hence the difficulty of finding one medicine to cure those with cancer. This means that, as far as we know, no single medicine can cure every type of cancer. Most of us know cancer as a terminal illness, but over the past few years, medicine has become incredibly advanced, and we may be looking at cancer becoming a chronic illness within the next 20 years. A chronic disease, such as diabetes, asthma, and depression, are conditions that don't have a cure, but you can live with and manage the symptoms. This has been hypothesized by many professors and oncologists, such as René Bernard, a professor of molecular oncology who has won the equivalent of a Nobel Prize in the Netherlands. Dr. Bernard stated that scientists have described the precise behavior of most cancer cells accurately. It is now to the pharmaceutical industry to develop the right medicines. In the mid-20th century, researchers came to a realization that to destroy cancer cells that had spread from the site of the original tumor, systemic treatments would be required. This treatment would differ from localized ones, such as chemotherapy and hormone therapy, in that the treatment would be able to travel through the bloodstream to reach and affect cancer cells all over the body. If, for example, breast cancer had spread to the brain, the medicine could also reach those breast cancer cells in the brain. This marked the start of the search for new and effective products to cure cancer. In 1955, the National Cancer Institute, the NCI, created the Cancer Chemotherapy National Service Center in response to a call for increased cancer screening and treatment programs. By 1960, the NCI expanded through a partnership with the U.S. Department of Agriculture to search for possible cancer cures from natural plant products. Between 1960 and 1981, researchers collected and tested an incredible 30,000 samples under this program. One of these samples proved to be immensely promising. In 1962, the botanist Arthur Barclay collected bark and other samples from the Pacific yew tree. 2 years later, two men from the Research Triangle Institute of the NCI, Monroe E. Wall and Mansuh Gawani, discovered that extracts from the Pacific yew tree were toxic to living cells. They then, with the help of their colleagues, isolated the most cytotoxic compound from the bark of the tree, and they called it Taxol. This was an extraordinary discovery, and its timing was perfect. Clinicians were struggling to treat cancer patients who were developing drug resistance or not responding to available treatment options. Taxol is proving to be a very promising discovery in cancer treatment. First seen as a miracle, the discovery of Taxol was soon realized to have complications. The slow-growing nature of the Pacific yew and the increasing demand for the compound led to the first roadblock. The drug was also very expensive to manufacture, and the ecological cost was just too high. Harvesting the bark kills the tree. To meet these challenges, researchers around the world raced to develop a synthetic form of the compound. In 1991, the NCI was able to produce a semi-synthetic, partly synthetic and partly natural in composition, form of Taxol, and in 1994, the Florida State University succeeded in the total synthesis of Taxol. This meant that Taxol could be produced in high quantities to cure patients from cancer. Today, Taxol remains one of the best plant-based cancer treatments available. This proves that nature can be an inspiration for scientific innovation. So, how does Taxol kill cancer cells? To answer this question, we first have to find out how normal cells divide and then if cancer cells divide in the same way. How do normal cells divide? Cells go through a life cycle, the cell cycle, which is divided into two major phases. The first, the interphase, and then the mitotic phase, or the dividing phase. In short, during interphase, the cell grows and makes a copy of its DNA. And during the mitotic phase, the cell separates its DNA into two sets and divides its cytoplasm, forming two new cells. Now in more detail, what exactly happens during the interphase? It is a state of preparation. This happens in 3 steps: the G1 phase, the S phase, and the G2 phase. During the G1 phase, the growth phase, the cell grows physically larger and copies organelles, which are tiny cellular structures, to prepare for DNA replication. In the S phase, the cell synthesizes a complete copy of the DNA in its nucleus, the control center of the cell, which contains a cell's genetic material. Lastly, during the G2 phase, or second growth phase, the cell grows more, makes proteins and organelles, and begins to reorganize its contents in preparation for mitosis. The G2 phase ends when mitosis begins. Now the cell starts to divide. The cell has reached the mitotic phase. In this process, known as mitosis, one cell, known as the mother, divides to produce 2 new cells, the daughter cells, which are genetically identical to the mother. Mitosis is essentially the part of the division process in which the DNA of the cell's nucleus is split into 2 equal sets of chromosomes. The goal of mitosis is to make sure that each daughter cell gets a perfect full set of chromosomes. Mitosis consists of 4 basic phases: prophase, metaphase, anaphase, and telophase. During the prophase, the chromosomes start to condense, making them easier to be pulled apart, and then the mitotic spindle begins to form. The spindle is a structure made of microtubules, strong fibers that form the cell skeleton. Its job is to organize the chromosomes and move them around during mitosis. Then, a part of the nucleus, the nucleolus, disappears, which is a sign that the nucleus is getting ready to break down. Soon, the nuclear envelope breaks down, releasing the chromosomes. During the metaphase, the chromosomes align at the metaphase plate with their kinetochores, a protein which serves as the attachment site for the spindle microtubules. This is called the spindle checkpoint and helps ensure that the cell was split evenly. During anaphase, the protein that holds together the sister chromatids, the 2 halves of the replicated chromosome, is broken down, allowing them to separate. Each is now its own chromosome. Lastly, in telophase, the cell is nearly done dividing, and it starts to re-establish its normal structures. The mitotic spindle is broken down, 2 new nuclei form, and the chromosomes begin to decondense and return to their original form. So, as mentioned before, cancer cells are cancerous because of the mutations present in the genes which control cell growth. However, as for the way in which they divide, cancer cells also undergo interphase and the 4 stages of mitosis. The only difference is that in cancer cells, the mechanisms that regulate systems, such as the one that makes cells proceed through the cell cycle, are defective. This means that these systems are activated regardless of these control mechanisms, and the system that normally stops cells from proceeding are inactivated. So, this means that cancer cells don't divide faster, but that they do not stop or take breaks between life cycles like normal cells do. They keep dividing. When looking under a microscope, cancer cells look very different from normal cells. They often have different sizes, are abnormally shaped, and the nucleus has a very odd appearance. So how does Taxol kill cancer cells? I will answer this question with the support of the research I did in the NKI lab, where I injected synthetic Taxol into a petri dish with breast cancer cells. This was then placed in a photo capturing microscope which took a photo every 10 minutes for 20 hours. These photos were then made into this video. How Taxol kills cancer cells is still an important question in the research field. As far as scientists are aware, Taxol works by stabilizing the microtubules which are found inside a cell's cytoskeleton, the network of interlinking tubules which play an important role in a cell's movement, shape, growth, and division. By stabilizing the microtubules, Taxol is restraining the cancer cell from dividing. Taxol also reduces cancer cells' dynamicity, their ability to change. This promotes the destruction of cancer cells by interfering with the cell cycle, leading to cell death. More specifically, it interferes with the spindle microtubule dynamics within the cell that ensures the correct alignment of chromosome pairs before they're pulled apart and become segregated into 2 daughter cells during mitosis. Microtubules need to be held at the correct tension for the chromosomes to align, for them to be able to divide. And anything that interferes with this process will ultimately cause cell death. The inability of the chromosome to achieve the phase in which the duplicated chromosomes are properly attached to the cytoskeletal structure leads to a mitotic block, in which cells cannot move forward in the cell cycle. They are stuck in a state of wanting to divide and being ready to, but not being able to. In the video, the brightness correlates to the length of this prolonged activation of the mitotic checkpoint. This can all be summarized by saying that Taxol induces mitotic arrest, in which a cancer cell cannot advance in the process of mitosis. This results in the subsequent triggering of apoptosis, cell death. Therefore, we see what appears to be the cells exploding. Taxol is primarily used for the treatment of breast, ovarian, and lung cancer, as it is proven to be the most successful in treating these types of cancer. There has been an improvement of nearly 40% in survival with good quality of life in patients with breast cancer that has spread, and a 39% 2-year survival rate in patients treated with Taxol, opposed to other medications with a rate of 20%. What are the limitations of Taxol? Taxol, as any chemotherapy, circulates throughout the body in the bloodstream, so it can treat cancer cells almost anywhere in the body. However, as is common for chemotherapy in general, Taxol does not know the difference between cancerous cells and normal cells. Although it is less likely to damage cells that are at rest, such as most normal cells, it affects healthy body tissues where the cells are constantly growing and dividing, such as hair, which is always growing, bone marrow, which is constantly producing blood cells, the skin and the lining of the digestive system, which are continuously renewing themselves. This results in low blood counts, mouth sores, nausea, diarrhea, and/or hair loss. Most side effects disappear once the chemotherapy with Taxol is over. Normal cells will be able to replace or repair the healthy cells that were damaged by Taxol. To get a better understanding of other limitations of Taxol, I interviewed Jonne Rijmakers at the NKI. What is the current research that is being done in regards to Taxol, and how can it be used to kill cancer cells?
Well, one of the major problems with Taxol in the clinic currently is resistance to Taxol, and people in research really try to understand why do tumors become resistant. And even though this drug has been used for years and years in the clinic, the resistant mechanism is still poorly understood. Another question that researchers are working on is how does Taxol kill cancer cells? So it has been long thought that it inhibits cell division. And indeed, if we do experiments in the laboratory, we indeed see that Taxol kills cells in dividing cells during cell division. However, the people also saw effects of Taxol in tumors that grow not so fast, so they don't divide so often, and yet they still die from Taxol. So this indicates that Taxol might also be able to kill cells that are not dividing, and this is now also something that has been tested more, or people are interested in research to see, is it indeed that Taxol only kills dividing cells, or can it also have an effect in interface cells that are non-dividing? And it seems to be the case that Taxol can also kill cells in interface. So the mechanism of Taxol, although it was long thought to be known, it now has some new implications.
What is the success rate of Taxol so far?
It has been used on patients a lot. It's one of the longest approved drugs, I think, in the cancer clinic. I think the cancer where it's used most for is breast cancer, but it's also applied for some other cancers. But in breast cancer, it's very, very effective, and even since 40 years, or I don't know how long it's available, but it's still one of the most effective chemotherapies for breast cancer.
In regards to how Taxol affects cancer cells, how is this treatment different or more effective than other treatments?
Well, Taxol is often used in combination with some other chemotherapies. So you have different types of chemotherapies. You have the taxanes, which actually kill cells in mitosis mostly, at least that's what we think. And you also have drugs that target the DNA replication machinery of cells. They bind to DNA and they damage the DNA. So that's a very different type of chemo. And by combining these two drugs, it has been shown that you get a maximum effect. So Taxol is not used a lot as a monotherapy, but often in combination, and this has proven very successful. It's not necessarily better, but it's often it has the maximum effect when it's used in combination with other therapies.
Where do you see Taxol as a method of killing cancer cells going within the next 50 years?
It's an interesting question. The development of drugs is very, highly evolved the last few years. But yet Taxol is still one of the most efficient drugs. So I think it will still be used in 50 years. We are now able to also produce Taxol-like compounds, not from plants, but also from in the laboratory, from chemical structures. And then it allows you to make very small differences or small adjustments to the Taxol structure, which might make it even a bit more efficient or longer binding. And so there's development of these type of drugs, but I think taxane-related drugs, they will still be used in 50 years.
In this case, nature was the inspiration for medical innovation. Taxol, originally isolated from the yew tree, can now be made synthetically and is effective in treating certain types of cancer, such as breast, ovarian, and lung cancer, when used in combination with other forms of chemotherapy to prevent resistant buildup. Research scientists are now trying to manipulate synthetic Taxol by making small adjustments to the molecule to target specific characteristics of cancer cells in a way that enables it to become even more effective. Finding new medications is often a path of trial and error or deduction. Taxol was discovered in the 60s and is still used as an effective treatment for certain cancers. The exact manner in which Taxol works is still not fully understood, which creates opportunities for further research. And this is what makes it so interesting.